Good morning, my dear students. Welcome to my channel Patel Science Academy. This is your Shahzad sir, and I welcome you in the chapter Inheritance and Variation, and it's 7 number lecture. And the 7 number lecture topic is chromosomal basis of inheritance. And in this topic, the point I'm going to explain you is the chromosomal theory of inheritance. So, dear students, what is this chromosomal theory regarding the inheritance? Actually, Gregor Johann Mendel published his work on inheritance of traits in 1866, but for some reasons it remained unnoticed or unrecognized till 19th century. So, dear students, as communications was not easy in those days, his work was not widely recognized. But his approach of using the mathematics and statistics to explain the biological phenomenon was totally new and unacceptable to the biologist. So, as continuous variations were observed in the nature, Mendel's concept of factors, that is the factor of the gene, as stable and discrete unit which control the expression of the characters, and that is a pair of allele, did not blend with each other and was not accepted by his contemporaries. So, dear students, he also did not know the physical location of the factors, that is nowadays it is called gene, in the gametic cell. So in 19th century, the three scientists, Hugo de Varais, scientist number 1, Correns, scientist number 2, and the von Schermark, the scientist number 3, independently rediscovered the Mendel's work on the inheritance of the trait, and due to the advancement in the microscopy, scientists were able to observe cell division and the structure of the chromosomes under the microscopes. So, dear students, this theory is regarding their observation. Walter Sutton, along with the Theodore Boveri in 1903, studied the parallel behavior of the Mendel's factor, that is nowadays called gene, and the behavior of the chromosomes at the time of the meiosis. So, dear students, based on these observation, chromosomal theory of inheritance was put forth by the Sutton and Boveri, and this theory identifies the chromosomes as the carriers of the genetic material. So, dear students, this is actually the chromosomal theory of inheritance, in which the chromosomal theory of the inheritance is an idea that the genes, the unit of the heredity, are physical in nature and are found in the chromosome. So this is regarding that gene, and it states that gene is the part of the chromosomes, and it is the unit of the heredity, and chromosomes are seen in the all dividing cells and are passed from generation to generation. These are the main postulation of the chromosomal theory. So now we'll see the structure of the chromosomes and its type in this lecture. So chromosomal theory, as you know, the chromosomal theory of inheritance is based on the few fundamental principles, and that I explained you earlier. Actually, based on these observation, chromosomal theory was put forth by the Sutton and Boveri, and the uh, theories identifies the chromosomes as the carriers of the genetic materials. So it is the conclusion of this theory that chromosomes act as the carriers of the genetic material, and gene is the small fragment of DNA and which is present on the chromosome. So this theory states that the chromosomes are present in a pair in somatic cells. During the gamete formation, homologous chromosomes pair segregates and assort independently. Assort means distribute independently during the meiosis type of cell division. Thus, each gamete contains only one chromosomal form of a pair. So, dear students, the nucleus of the gametes contain chromosomes which carry all the hereditary traits. Male and female gametes, that is sperm in the male and egg in the female, All these gametes carry all the hereditary traits, and they are the link between the parents and the offspring. The fusion of the haploid male gamete and the haploid female gamete, that is sperm and the egg, it restored the diploid number of the chromosomes of that particular species. So this is the conclusion of the chromosomal theory, which includes: chromosomes contain the genetic material. Chromosomes are replicated and passed along from parents to the offspring. The nuclei of the most eukaryotic cells contain the chromosomes that are found in the homologous pair. During the meiosis cell division, each homologous pair of the chromosomes segregates or separates into one of the two daughter nuclei, and during the formation of the gametes, different types of the non-homologous chromosomes segregates independently. Each parent contributes one of the sets, that is sperm from the male and egg from the female, to its offspring, and the set are functionally equivalent, and each carries the full complement of the gene, means the diploid number of the chromosome. So this is the conclusion of this chromosomal theory. Now we'll go, before we understand about the chromosome, we have to just learn about the ploidy and its type. What is ploidy? Actually, as I told you, chromosomes are filamentous body present in the eukaryotic nucleus in the eukaryotic cell. The term chromosomes, it is made up of two Greek word, that is chroma and zoma. Chroma means color and zoma means body. So the word to word meaning of the chromosomes means the color body, which is genetic material present in the nucleus of the eukaryotic cell. It was coined by the Valdier in 1888, and the size of the chromosomes varies from species to species. Each metaphase chromosomes varies from 0.1 to 33 mm in length and 0.2 to 2 mm in thickness. Chromosomes are visible during the cell division, and they are capable of self-replication and play the vital role in the heredity, mutation, variation in evolutionary development of the eukaryotic species. So dear students, chemically eukaryotic chromosomes are made up of DNA, histone protein and non-histone protein, and the function of the chromosome is they are responsible for the for carrying of heredity, means they are the source of heredity. They are used as a carriers of heredity from parents to their offspring. But what is euploidy, or what is ploidy? The number of the sets chromosomes in the cell. So as I told you, chromosome is a genetic material present in the nucleus of the cell. And what is ploidy? The number of the sets of chromosomes in the cell. How many sets of chromosomes are present in the cell? That is called its euploidy, or in the cell of an organism it is called ploidy. So ploidy means the sets of chromosomes in the cell. So dear students, it is totally about the number of the chromosomes. The number of the chromosomes is specific and constant for a particular species. Therefore, it is of great importance in the study of the phylogeny and taxonomy of the species. The term ploidy, as I told you, speaks for the degree of the repetition of the primary basic number of the chromosome, that is X, in a cell. When the chromosome number in the cell is the exact multiple copy of the primary basic number, then it is called euploidy. So everything about the ploidy and its type we'll see in the next slide one by one. So dear students, in these slides you have to just remember ploidy means the number of sets of chromosomes in the cell. So this is about ploidy. Now we'll see about the ploidy. Now we'll see about the ploidy, heteroploidy, types of heteroploidy, euploidy, its definition, types, its subtypes, example, origin and change in the euploidy, and their causes one by one. So let's see it in a detail. What is called euploidy? Euploidy is the state of the cell. It is the state of the cell or organisms having one or more than one set of the same set of chromosomes. So dear students, as I told you, ploidy speaks for the degree of the repetition of the primary basic number of the chromosomes in a cell. When the chromosome number in a cell is the exact multiple of primary basic number, then it is called euploidy. So euploids includes the monoploids, that is haploid, with one set of chromosomes. Haploid means monoploid, only one set of chromosome is there, which is indicated by small n. Then after monoploid there is diploid. Diploid means diploid means two sets of chromosomes, so it is indicated by 2n. Then triploid, then triploid means three sets of chromosomes, it is indicated by 3n, small n is there. Then tetraploid means, tetra means 4, so 4 sets of chromosomes, it is indicated by 4n. So likewise, these are the number of chromosomes as far as euploidy is concerned. So euploidy means the state of the cell or organism having one or more than one set of the same set of the chromosome, possibly excluding the sex chromosome. Sex determining chromosomes means about the autosomes. So for example, we'll try to learn about the euploidy here. Most human cells have two of each of 23 homologous monoploid chromosomes for a total 46 number of chromosomes, and a human cell with an extra set of 23 normal ones would be considered as a euploid. For example, euploid karyotypes would consequently be the multiple of a haploid number, which is in human 23, and single extra chromosome such as the Down syndrome, it will be 24. So karyotypes are given names with the suffix ploidy used for the euploid karyotypes. So dear students, in the euploid we can consider here one of the type of the euploid indicated by small n means having one set of chromosomes. Then one set of chromosome, it is called haploid. Then two sets of chromosome, it is called diploid. Then three sets of chromosomes indicated by 3n, it is called triploid. And then four sets of chromosomes, it is indicated by 4n, it is called tetraploid. Then 5 sets of chromosomes, pentaploid. So likewise, these are the different types of the euploidy in which there are different multiple sets of chromosomes are there. If one set is there, it is indicated by small n and it is called haploid. If two sets are there, it is indicated by 2n and called diploid. If three sets are there, it is called triploid indicated by 3n, and if four sets are there, it is indicated by 4n and it is called tetraploid. So this is about euploidy and its type. Then what is aneuploidy? So as you know about the euploidy, now we'll see about the aneuploidy. Most common cause is non-disjunction of either pair of homologous chromosomes during the meiosis 1 or the failure of the sister chromatids to separate during the meiosis 2. So the result gametes will have either one less chromosomes or one extra chromosome. So dear students, as I told you in the previous example, we have seen about the euploidy. In the euploidy, if single n is there, that will be called haploid. If 2n is there, that will be called diploid. So I'll explain you about the aneuploidy. For example, we just take the example of human being. In the human being, haploid cell means n. Haploid cell contain 23 number of chromosomes, and diploid cell, that is 2n, it contain full number of chromosome, that is 46 number of chromosomes. So it is normal euploidy. This is haploid number, this is diploid number in the human being. Haploid number is 23 and diploid number is 46, half of this diploid number. But dear students, what is aneuploidy? Aneuploidy means the resultant gametes, because of the failure to separate during the meiosis, what will happen, this diploid cell will not be able to convert into its normal haploid cells because of the failure of the separation during the meiosis. So what will happen, instead of 23, either it will have 22, one chromosome less, or 24, one chromosome extra. So this type of gamete which will have abnormal number of chromosomes in the set, as I said, in the cell of that organisms, that is called aneuploidy. So as this 23 number of chromosomes in the set, it is having one chromosome less, so this is called aneuploidy. And again the another example in which one extra chromosome will be there, instead of 23 it will have 24 number of chromosomes, so it is called again aneuploid cell. So this is the difference in between the euploid and aneuploid cells. Now we'll go for the next slide. So till now we have seen about the ploidy, the number of sets of chromosomes in the cells. It is divided into two types, euploidy and aneuploidy. So euploidy may be monoploidy, means haploid, indicated by single N. It may be diploidy, indicated by the double N. It may be triploid, tetraploid, pentaploid, means polyploid, more than 2. So this is called euploidy, indicated by N, single N, and for monoploidy or haploidy, and it is indicated by small n. So this is about the euploidy. And what about the aneuploidy? Aneuploidy may be hypoploidy or hyperploidy. Hypo means less and hyper means more. So hypoploidy will have one chromosome less, as 23 in the cell instead of as 22 in the cell instead of 23, and hyper means more, it will have one extra chromosome, instead of 23 it will have 24 number of chromosomes in that gamete. So this is called hypoploidy and hyperploidy. Then polyploidy may be autopolyploidy and allopolyploidy, that we'll see later. And hypoploidy may have monosomy and nullisomy. In monosomy and in nullisomy, what will happen, I'll explain you. Aneuploidy is either addition or deletion of one or more chromosomes to the total number of the chromosomes in a cell, that as I have explained you. So dear students, now we'll see here in this example, ploidy is divided into euploidy and aneuploidy. Euploidy having the number of sets of homologous chromosomes. It may be monoploid, having single end, means 23 number of chromosome in the human. It may be diploid, having 2 number of chromosome, means 46 number of chromosome in the human being. Then it may have 3n, 4n, likewise, that is called polyploidy, more than 2n, means polyploidy. It may be autopolyploidy or allopolyploidy. But what about the aneuploidy, which is divided into hypo and hyperploidy? In the hypo, it may be monosomy and nullisomy. In the monosomy, there may be one chromosome less than the normal, and in the nullisomy, there may be two chromosome less than the normal gametes. Thereafter, in the hyperploidy, there may be trisomy condition or tetrasomy condition. In the trisomy condition, one chromosome is extra in the gamete, that is 23 plus 1, that is called 24. And in the tetraploidy, two chromosome may be extra in number. So this is about the ploidy and their number. So here we can see this is ploidy divided into euploidy and aneuploidy. Euploidy may be haploidy, means monoploidy with a single N, diploidy with the 2N, and polyploidy may be with the 3N, 4N, 5N, etc. and so on. Likewise, the aneuploidy may be hypo and hyperploidy. In the hypo, one chromosome may be less and two chromosomes may be less. If one is less, it is called monosomy. If two is less, then it is called nullisomy. Then in the hyperploidy, one chromosome may be extra or two chromosome may be extra. If one chromosome is extra, it is called trisomy, and if two chromosome is extra, it is called tetrasomy. So these are the variation in the chromosome number, that is called ploidy, and this is the tree diagram of ploidy. I hope you have understood about it. Now we'll see about the chromosomes. In the what is chromosomes? As I told you, dear students, it is the Greek word and made up of two words, chroma and soma. So this is that two word, chroma and soma. Chroma means color and soma means body. So chromosome means color body, and it is found where? It is found in the nucleus of the each cell, each eukaryotic cell. The DNA molecules is packaged into the thread like structure that is called chromosome. So DNA is the part of the chromosome, and each chromosome is made up of DNA tightly coiled many times around proteins that is called histone proteins, and that supports the structure of the chromosome. So chromosome is a color body, it is the genetic material, and it function as a carriers of heredity, and it is it this the size of the chromosomes, it varies from species to species, and it is coined, means it is first time, this term first chromosome first time used or coined by the W. Valdier in 1888. So each metaphase chromosomes, so it is visible during the metaphase under the microscope, and it varies from 0.1 to 33 μm in length and 0.2 to 2 μm in thickness. So these chromosomes are visible during the cell division, that is metaphase stage. So this is about the general information of the chromosomes. Now we'll see the structure of the chromosomes. So dear students, this is the structure of the chromosomes. The chromosomes are best visible under the microscopes when the cell is at metaphase stage. So question may ask, when you will be able to watch the chromosomes in the cell during the during the meiosis cell division. So it is always metaphase stage, and during the mitosis and the meiosis cell division. So metaphase stage is that stage during which chromosome is visible, and in it is because at this stage chromosomes are highly condensed, and typical chromosomes, actually dear students, consist of two chromatids joined together at a centromere or the primary constriction. So are you observing here two chromatids in this structure? This is one chromatids and this is another one chromatids. So these chromatids are joined together by this round shaped structure present at the middle, and this is called centromere. So dear students, these chromosomes consist of two chromatids, as you can see here, and they are joined at the one point. That point is called centromere or primary constriction. So this centromere, it consists of a disc shaped plate, and that is disc shaped plate is called kinetochore. So dear students, you can see here this structure. So the disc-like shape plate is of the primary constriction, that is centromere, is called kinetochore, and it is at the kinetochore the spindle fiber get attached during the cell division, And besides this primary constriction, which is also called centromere, some few chromosomes possesses additional one or two constriction, that is called secondary constriction. You can see here, and at the secondary constriction, one nucleolus become organized during the interphase, and a satellite body, that is SAT body, which is called satellite body or SAT body, it is attached as a secondary constriction to in very few chromosomes. Each chromatid in turn contains a long, unbranched, slender, highly coiled DNA thread like structure, and that is called chromonema. And this chromonema extending through the length of the chromatids. So this chromatid consists of a double stranded DNA molecule which extend from one end of the chromosomes to other. So dear student, this is that structure. In this structure you can see different part here. This structure in this chromosome is called chromonemata. This middle part is called centromere. We joined these two structure with each other, and inside this there is a colorless liquid that is called matrix, and this is actually the matrix nucleolus. And this is that primary constriction which is called centromere, the middle of the chromosomes. This is chromonemata. This is secondary constriction number 1 or nucleolar organizer. This is satellite. This is nucleolus. This is secondary constriction 2. This is secondary constriction 1. This is secondary constriction 2. This is chromomere, the coiled structure, and the uppermost part of this chromosome, this is called telomere. So this is about the chromosome structure in short. The homologous chromosomes, sister chromatids, centromere, all these things are there. As you can see in this structure, this A part, it indicates the chromosomes. This B part, this is the central part, it is called centromere. These two chromatids are there, this is called sister chromatids as a C, and D in this diagram is indicating the homologous chromosomes which is having the same structural part. This is about the structure of the chromosome. One more time we can see here the structure of the chromosome. In a chromosome there is a satellite. You can see from this lowermost part to uppermost part, satellite is there. Then secondary constriction number 1, secondary constriction number 2 is there. At center there is a centromere, satellite is there. Then secondary constriction number 1 is there. This is secondary constriction number 2 is there. Then at center centromere is there. We joined these two structure, and these are the chromonemata. This is chromomere, and at the uppermost part there is telomere. So this is about the structure of the chromosomes. The types of the chromosomes on the basis of location of the centromeres. So dear students, on the basis of the centromere there are 4 types of the chromosomes, that is called, you can see this is the centromere which joins these two sister chromatids with each other, and on the basis of this centromere, which is also called primary constriction, on the basis of that centromere this chromosomes is divided into 4 types, and these 4 types are likewise. So you can see here the 4 type of the chromosomes. What are you observing here? Centromere at center. This is centromere some distance away from the center. This is centromere about nearer to the tip of the chromosomes, and this is centromere at the tip of the chromosome. So on the basis of place of or position of the centromere. These are the different four types of the chromosomes. If centromere is located at the center, so it is called metacentric chromosome. If centromere is located some distance away from the center, this is called submetacentric chromosomes. If this centromere is nearer to the tip, it is called acrocentric chromosomes, and if this centromere is present at the tip, it is called telocentric chromosomes. So these are the four types of the centromere. These are the four types of the these are the four type of the chromosomes on the basis of the position of centromere. So dear students, this is about this four types of the chromosomes. Four types of the chromosomes, in which chromosome is called metacentric, in which chromosome is called metacentric if centromere is present at the middle or at the center of the chromosomes, and during the cell division this metacentric chromosomes appear as a V shape. Then submetacentric chromosome. During this submetacentric chromosomes, what will happen? This centromere is located some distance away from the center. So this is not the center of the chromosome. This is the centromere present some distance away from the centromere, and this is called submetacentric chromosomes. And these submetacentric chromosomes, it appears L shape during the cell division. Then acrocentric type of the chromosomes, in which this centromere is located nearer to the tip. As you can see, this is the tip of the chromosome and it is located nearer to the tip, and this type of chromosome is called acrocentric chromosome, and this acrocentric chromosome, it appears J shape during the cell division. And then the last type of the centromere, it is called telocentric. Last type of the chromosome, it is called telocentric, as the centromere is present in this structure, in this type, at the tip of the chromosomes, and it appears as a rod shape or eye-shaped structure during the cell divisions. So these are the four types of the chromosomes on the basis of position of centromere, that is called metacentric, submetacentric, acrocentric and telocentric. This is about this lecture. Thank you for watching this video. Dear students, please do subscribe my channel Patel Science Academy on the YouTube, and share my videos with your friends and with all the persons who want to learn about this topic, and uh, press the bell icon so that you can receive my instant notification of the new video. Thank you very much for watching this video.